My name is Michael Baum, and I'm the team leader for breeding and scaling of cereals and legume varieties. And I'm also the research platform manager for Morocco. I'm working for ICARDA, the International Center for Agricultural Research in the Dry Areas, with headquarters in Lebanon. At ICARDA, we are in the field I'm responsible for is the breeding of dryland crops. We have a total of 7 breeding programs, which is wheat, barley, chickpeas, faba bean, lentils, and laterals. We have 3 research platforms, which we use to test for the breeding and to test options before we outscale them. The 3 research platforms are in Morocco, in Turbol, and in Amlaha, India, respectively. Our breeders, they use conventional breeding programs to develop new material, which are tested with the national programs in the dry areas, reaching from Morocco to India, Pakistan, and China, in national trials. And the best materials will be promoted as new varieties. One area under breeding is the area of biotechnology, which provides very important tools to make the breeding more efficient and more precise. For instance, one area is the application of DNA molecular markers. That starts with the molecular characterization of germplasm from the gene bank. For instance, ICARDA gene bank hosts 143,000 accessions of our mandated crops, and a large amount of these accessions are characterized with molecular markers in order to identify redundancies and duplications, and but also to help us to understand or to identify the rare alleles that we want to use in the breeding program. Within a conventional breeding program, molecular markers are used for what we call genomic selection. The breeder in the past used to rely entirely on his eye and his hand to select superior varieties. But today we have a number of tools that help the breeders to do this selection even better than the eye and the hand. So these are, for instance, the molecular markers. So with the help of molecular markers, we can do parts of the selection based on breeding values rather than phenotypes. So basically the genotype of a variety is being selected with the help of molecular markers, which makes this a more precise exercise. Another important area where the breeding is supported by biotechnology is the area of genetic engineering, and nowadays specifically the application of genome editing using the CRISPR-Cas system. ICARDA has been developing and adopting plant transformation systems for its mandated crops in wheat, in barley, in lentils, and in chickpeas. And these systems, transformation systems, are important and needed when you want to use the CRISPR-Cas system today for genome editing. And so we are fortunate that we have these systems available. And here in Egypt, at the Agricultural Genetic Engineering Research Institute, AGERI, we have set up a group that specifically interested and concerned about using the genome editing approaches to improve our crops. And as I said, I mean, that's done on the mandated crops: wheat, barley, lentils, chickpeas, and faba bean. Of course, we have a specific interest in heat and drought tolerance, because ICARDA is the international center for the dry areas. So we try to develop germplasm that with resistances for abiotic stress, like heat and drought, and especially in times of climate change, this becomes very important. And but also a number of biotic stresses, which have been very difficult to modify with conventional breeding, can be now addressed with genome editing. One example I like to mention is in Laterus. It's an important legume crop in the highlands of Eastern Africa, Ethiopia, and in the highlands of Nepal, Bangladesh, and India. This legume is a miracle crop because it's one of the most drought-tolerant legumes, but it also withstands waterlogging because it has a deep rooting system. This crop has one problem. It has a neurotoxin in the seed called beta-ODAP. So if the population eats too much of it, it can cripple the lower limbs. For that reason, for a number of years, Laterus was forbidden. There was a 5 years moratorium on growing Laterus in India, which has been lifted again. But now with the CRISPR-Cas system, with the genome editing, it's possible to modify the genes that are responsible for the production of this neurotoxin. And already in a collaborative project with the John Innes Institute in UK and the University of Queensland, lines have been developed with zero beta-ODAP, with zero toxin. And these are some of the hopefully beneficial applications of the technology to help the population in the poorer countries of this world. One important activity of our work is capacity building and training. The research that we do, we don't do this only for within our research projects and for ourselves, but we have the tasks to also train the next generation of plant breeders. I mean, to train the plant breeders of the future with all the conventional as well as the modern technologies, such as the use of DNA molecular markers, the use of genome editing to deliver to the national programs new varieties, more precise and more faster than we were able to do in the past.